In this video, I want to talk a little bit about general bacteriology. This should be a review of Bio 120. If this material seems really new to you, you probably should go back and refer to your Bio 120 notes to refresh yourself in general bacteriology. That said, we'll probably move through these slides really quickly, but of course you have the slides, you have the video, and you can watch them and look at them at your own pace. In general, when we talk about bacteria, in a medical microbiology class, we don't talk about archaea. Sometimes we do use the term prokaryote, but really in this class, we're only talking about bacteria. Bacteria don't have a nucleus. That's what you can see in this image here. The chromosome is generally supercoiled and circular and floats around within the cell. Some bacteria do have two chromosomes, for example, the genus Vibrio, which we'll talk about later, but for the most part, it's a single circular chromosome. Some bacteria also have linear chromosomes like Borrelia. We have peptidoglycan, which is the major component of the cell wall, a cytoplasm with 70S ribosomes. Some bacteria will have plasmids, some will have flagella. They have a cell membrane, some have two. Not all bacteria have a cell wall, most do. We'll talk about the mycoplasma, which don't. But these are the general features of a bacterium, whereas eukaryotes are different. They have more complex organelles, including a nucleus, which is where the DNA is. They have mitochondria for respiration, lysosomes, endoplasmic reticulum, Golgi. They also have different ribosomes, and that becomes important when we talk about antimicrobial or antibacterial therapies. Again, here's your chart that compares eukaryotes, which have a nucleus, and prokaryotes or bacteria, which don't. When we start to talk about bacteria, we also classify cells as gram-positive or gram-negative. Again, I don't think I need to get into too many of the details because you should have already covered them. But important features that distinguish the two that will become relevant when we talk about symptoms and treatments include the presence of things like teichoic acids in gram-positive cells, a single cell membrane in gram-positive cells, the presence of LPS and its various components, including lipid A, which serves as an endotoxin in gram-negative cells, and the fact that they have two membranes. All of that becomes relevant when we talk about treatment and how they cause disease. So when we talk more about the specific organisms, we'll talk more about why those particular things are important. The other image shown here is a cross-section of an example of a mycolic acid-containing cell surface or a mycobacterial cell wall. You see it's sort of organized like a gram-positive in that it has kind of a thicker layer of peptidoglycan. It has a single membrane. However, it then has this covering of mycolic acids, and they're very waxy. And that prevents them from being stained like a typical gram stain. You have to use an acid-fast stain. So as part of your reflection and part of the thing you need to think about the differences between gram-positive and gram-negative cells, what the implications might be for symptoms, how they stimulate the immune system, and also for treatment of these types of infections. We will talk about two genera of bacteria that have the mycolic acids as well. So we have the gram stain shown in the top image. Again, you should be familiar with this. You should have already done it. You should know what it means and why it's important clinically. And then, of course, the different morphology of cells, which can be helpful for those of you in the clinical laboratory sciences when you're doing microscopy to be able to identify basic morphology and also how the cells are arranged. Are they in clusters? Are they in chains? Are they individual? Things like that. Various internal and external bacterial structures are shown in this image. On the left-hand side is an example of a gram-positive cell. On the right is a gram-negative cell. Not all gram-positives have all the features shown. Not all gram-negatives have all the features shown. But they're trying to show you what classic things you could see. So some of the optional features, I guess bonus features, are the ones that are in parentheses, like capsules, flagella, and pili. The other features are generally seen in almost all cells. Again, you have a chart that you can refer to at any point that shows you some of the structures of gram-positive cells, some of them of gram-negative cells. You will see some overlap. Gram-positive and gram-negative do have some similar characteristics, but also they have some differences. And those differences can be highlighted in this chart as well. So this is something that you can refer to see how these cells are different and how they're the same. Any cell that has to make a cell wall, which is almost anything except mycoplasma, put together two subunits of a carbohydrate. N-acetylglucosamine is the one on the left, N-acetylmuramic acid is the one on the right, and then a chain of peptides, alanine, glutamate, lysine, alanine. And why I'm showing you this is because, one, you can see right here lysozyme cleavage site. So lysozyme, which is an antimicrobial enzyme produced by our bodies, can cleave peptidoglycan. So you can think about what would the implications be for lysozyme helping us to fight infections. And then one of the other things to think about is where various antibiotics that inhibit cell wall production might act. And some of them can prevent this cross-linking. Some of them can interfere in other ways. They can be incorporated instead, which causes the cell wall to fall apart. So I'm not going to ask you all of the details of how the cell wall is put together, but just seeing it and seeing where antibiotics could act or where our own body's defenses could act hopefully gives you an idea of how we can fight or treat infections. The LPS, which again is found only in gram-negative cells, is a component of the outer membrane. It's made of an O antigen, a core polysaccharide, and this can be different depending on which species we're talking about, but some of the components of the core polysaccharide are shown here, and some of the components of the O antigen are shown here. So this can sometimes differ between species. We'll see some species of bacteria have what's called an LOS, so a lipo-oligosaccharide instead of a lipopolysaccharide, because they have fewer repeating units of the O antigen. And then we also have lipid A, which serves as the endotoxin. This is the part that stimulates the immune system. And again, we call it endotoxin because it's an actual component of the cell rather than something secreted by the cell, which would be an exotoxin. And then here's a more detailed diagram of what lipid A looks like. This is just a cartoon showing cell division in a bacillus-shaped organism. Again, I'm not going to ask you tons of the details about this. You've already seen it. You already know it. It's just part of general bacteriology. One special feature, maybe, or different type of a modification of cell division is the formation of a spore. The technical term would be endospore to differentiate it from like fungal spores, but it's okay if you just call them spores. So spores are resilient structures produced by some genera of bacteria. Almost all of them are gram-positive. Some of them will stain gram-negative, but technically they're organized like a gram-positive, so it doesn't really matter. We will talk about two medically relevant genera that produce endospores. Those would be Bacillus, and the major disease-causing organism we'll talk about is Bacillus anthracis, which causes anthrax. And then Clostridium. The two relevant species that most people are aware of would be Clostridium botulinum, which can cause botulism, and Clostridium tetani, which can cause tetanus. The spores are really important to help these genera survive harsh environmental conditions, including nutrient limitation, dehydration, UV radiation, any sort of stressor that the bacterium might face can cause them to sporulate or form a spore. Once the spore has formed, sometimes those spores can persist in the environment for literally millions of years. So these are really, really resilient structures. When conditions improve, they'll become vegetative, and that takes about 90 minutes. And so you would go from having a spore to having a vegetative cell.